see. Yep, I am recording. All right, any questions just in general about the class before we begin? I know it is a lot of content. I got that feedback from your content checks. I understand this is actually less content than the last time I taught it. So I have pared down as much as I possibly can while still being able to give you the content I need to teach. So I know it's a little daunting though. All right, so today we're going to continue with talking about our cells. So on Monday we talked about a lot of the different organelles, and we're going to continue with our organelles today. But we're going to start with our semi-autonomous organelles. Right? So they're organelles, but they're just slightly different in how they originated and how they act. So two of these organelles would be mitochondria and chloroplasts. Right? These two organelles, they will grow and reproduce themselves, and they contain their own DNA. Now, they're not completely autonomous, right? They can't essentially live outside of the cell because they still rely on the cell for some materials, right? So, for example, some of the molecules that it needs to make proteins inside of these organelles might come from what's being produced by the rest of the cell. So it still relies on that cell. Right? If we talk about our mitochondria, right, you might have often heard the phrase, the powerhouse of the cell. And yes, we are going to talk a little bit more about that on our next unit when we talk about cellular respiration. But mitochondria and chloroplasts, their job is to convert the energy given to them into a type of energy that is usable by the cell. Right? So for mitochondria, this would be in the form of cellular respiration. Right? It's taking in those food particles, those macromolecules that we are giving our bodies and changing that into a form of energy that the cells can use. And this energy form is ATP. Right? We find mitochondria in all plant cells and in all animal cells. Now, chloroplasts, they're only found in plant cells. I might amend that a little bit and say any photosynthesizing cells. Right? So algae, not quite plants, but they can contain chloroplasts as well. These chloroplasts, like I said, they're photosynthesizing. They're taking in energy in the form of sunlight and converting that energy into a usable form for the cell into sugars. Right? If we look at our mitochondria structure here, you can see that we have all these little folds on the inside here, and inside all of our folds, inside of these membranes, would be called the mitochondrial matrix. And it's this matrix that hosts or that holds the DNA. Right? We call it mitochondrial DNA because it's DNA specific to the mitochondria. But they also have ribosomes, and they also have a variety of enzymes used for cellular respiration. Right? And again, you will see mitochondria in plants and animal cells. Now our chloroplasts kind of look similar in structure in that they have a lot of membranes on the inside. Right? These membranes are used to carry out photosynthesis, but the fluid surrounding all of our membranes here is what is called the stroma. Right? It's a really thick fluid. It contains the chloroplast DNA. Again, we're calling it chloroplast DNA because it's specific to that organelle, ribosomes, and a variety of enzymes for photosynthesizing. Right? So in a way, these two organelles are very similar. They perform similar types of functions, right? But photosynthesizing versus cellular respiration is the big difference there. Right? These cells are able to reproduce on their own, right? So they have their own set of DNA. Right? We can see that in this image here. This DNA is this little red blob on our image here. All right? We have our mitochondrial DNA. When these cells divide, the same thing happens in chloroplasts here. When they divide, they're going to replicate their own DNA. Okay, we can see that in this next step here. Now we have 2 of our red dots. Okay. Once the DNA has been replicated, the cell will then divide in 2, and now we're left with 2 mitochondria. All right. Very much a simplified version of what's going on here, but know that they can reproduce on their own. And the purpose of reproducing is that a cell might need more mitochondria. Maybe the cell has grown, right, and it has more energy needs. So we need to add in more of these mitochondria to the entire cell. Maybe this cell is getting ready to divide and go through mitosis or meiosis, right? It needs to replicate all of its organelles. And mitochondria would replicate in this fashion. Same with chloroplasts. All right, I want us to take a step back and maybe relate to the homework I had you do here and answer the question, why do mitochondria and chloroplasts contain their own DNA? All right. I'm going to give you about 2 minutes to discuss with each other and ask me questions. Anyone else printed the assignment that was due today? I will take it. You can drop it off before you leave class. Otherwise, there was a submission online. But because it is quiet, let's move on. Why do mitochondria and chloroplasts contain their own DNA? About particularly one of those videos I had you watch. Yeah. So we theorized that mitochondria and chloroplasts, they were prokaryotic cells, right, living their lives, but they're able to convert all these different sources into an energy source. And then a larger eukaryote came along, kind of ate it or absorbed it, however that original eukaryotic cell took in food particles. And then it became beneficial to that cell and they lived together. Right? So we think it had its own DNA because it used to be its own organism. Does that make sense? All right, and that's what I have on my next slide here. Right, I'm not going to talk about it too much because I had you watch that video here, but it is this symbiosis theory, right? The theory that they originated from bacteria, right? They lived outside of the cell, and eventually became engulfed by a eukaryotic cell that kept it around because it was beneficial. Right? So endosymbiosis, right, which is the term I have missing up there for you in your slides, is this symbiotic relationship with a smaller species living inside a larger species. This is not the only example of endosymbiosis. Right? You can see endosymbiosis in other organisms today. For example, you might think about like bacteria that might live inside of you. Right? They're small, living inside of a larger species. All right, any questions on endosymbiosis? All right, So at this point, we have talked about a lot of components of the cell. Right? We have talked about the cytosol and the cytoskeleton, right? The structure that makes up the inside of our cell here. We have talked about the endomembrane system, including the nucleus. Right? So all those organelles contained inside of the cell. And then we just covered our semi-autonomous organelles like mitochondria and chloroplasts. And know that all of these organelles and structures inside of our cells, they all work together, right? So we talk about them as very isolated things, right? But they're all interconnected. I do want to mention very briefly that outside of the plasma membrane, cells can also have extra material, right? So in animal cells, we would call this extra cellular matrix. In plant cells, it's a cell wall. When I was taught this, my professor kind of explained it to me as like a sweater analogy, right? You have a sweater wrapped around your body, just like these wrap around our cells. Our sweaters are made of like these interwoven fibers, right? And that's very similar to the structure of our extracellular matrix and our plant walls. They're made of these fibers that are woven together to provide some rigidity to our cells. Right? So in animals, it's the extracellular matrix, secreted outside of that plasma membrane. Right? So we have the organelles on the inside, the extracellular matrix on the outside. It can play a variety of different roles. Right? I will tell you that I'm not going to ask you for these specific roles, but I do want to just explain them to you. They can provide strength, right? So skin and cartilage, they have really thick extracellular matrices. Structural support, bones are a great example of this if you've ever seen. A slide of a bone cell has a really thick extracellular matrix. It can help with organization. So some proteins might bind to the extracellular matrix, or it might help cells arrange themselves, as well as helping with cell signaling, right? So signaling between different cells. Now, some animal cells can have really thick extracellular matrices. Some of them might be barely present. Depends on what the cell is specialized for. Right? The same goes for these plant cell walls. Right? So plant cell walls, they provide this rigidity and strength. Often if you see an image of a plant cell, it's very rigid and kind of square-ish looking. Right? It's that cell wall that provides that shape to it. It's really good at resisting compression because of that, right? Which is great for plants because plants like to wilt. Right? You know, when plants wilt, that disrupts their plasma membrane a bit, but if they have these cell walls, that plant will stay upright until the plant eventually will die from lack of water. Really good examples of plant cells that have these thick plant cell walls are cells that make up the woody material of plants. Another great example is if you've ever eaten a pear and it's kind of gritty, right? That's because the cells in our pear, they're special cells called sclerids, but they have a really thick cell wall, which makes them feel gritty when you eat them. All right, so back to cells here. I just had that quick aside. We know we have all these different types of cells within our bodies. Right? On our image up here, I have a cheek cell that looks very different from a neuron. Right? Now, these cells contain exactly the same DNA, so what makes them so different from each other? And the answer to that are proteomes. Right? So proteomes are a set of proteins that are expressed. So all cells have the same DNA that encodes for the same proteins. But not all of those proteins will be expressed in every cell. Only a subset of those proteins will be expressed. Right? And it's a subset of those proteins that are expressed that lead to these very specific roles that cells can have. Right? So muscle cell versus a neuron versus a cartilage cell. Right? So the proteome is a set of proteins expressed. And when I say expressed, they are the ones that are actually physically made. Right? They can change in their level of expression. Right? So some cell might say express a lot of the specific type of an enzyme, whereas another cell might express that enzyme, but not in such a great amount as that other one. Right? So the amount by which they are expressed, which subtypes of proteins are expressed. So when I'm talking about subtypes, I'm talking about signaling proteins or enzymes, like different classes of proteins, right? So they might express, maybe they're only expressing different types of enzymes. Maybe they're really good at signaling proteins. So neurons, they like to signal. They would express a lot of signaling proteins. And finally, they can differ based on how those proteins might be modified after they are made. So whether or not, say, an extra carbohydrate was attached to the protein. Right? This can be relevant for disease, right? So on cancerous cells, the proteomes of those cancerous cells are really different from the proteomes of the healthy equivalent of that cancerous cell. So there's a lot of study that goes into this, but I'm just touching on the very basics here. All right, I want us to put together everything we've learned from today and on Monday, so you might have to go back in your notes, and fill in this table. So, my question is that you have harvested cells from different organs of a frog, right? Muscles, pancreas, intestine, and testes. When you view these cells under a microscope, you notice that they have variable numbers and amounts of different types of organelles depending on their function. Right? So now I have given you a table of the cell type and their function, and I am asking you what organelle might be most abundant in those cells based on its function. Right? I'm going to give you like 5 minutes to work through this because I know it's a little bit of a heftier lift for this question. Great time to review notes, great time to ask some questions. cargo. I can see where you're going with it, though. And then we have a question about the testy cells. Yeah, so what are steroids? What's more classes of biomolecules we talked about? Yeah, we talked about four in total. Now, carbohydrate, what the heck? What about the class of biomolecules? We said we had proteins, we had carbohydrates. We also talked about lipids. Yes, lipids. The last one being those nucleic acids. For pancreas? There's a lot that could play into this pancreatic cell. There will be multiple answers that expect. Depending on your context, I am understanding in those things. And then I get your answer, and I'm like, oh yeah, that's definitely yeah, this is definitely one of those questions with like a little bit of complication for multiple things together. All right, looks like our 5 minutes is up here. So if I could ask for volunteers to give me their answers, pancreatic cells, we synthesize digestive enzymes. What organelle or organelles might be really prominent in this cell? Yeah? Yeah? I would accept ribosomes because we're synthesizing proteins. Enzymes are proteins. I would accept rough ER because that's specifically where the ribosomes are attaching and synthesizing the proteins at. I would also accept lysosomes because lysosomes, they carry the digestive enzymes. Right? So I would accept the 22 different answers there. What about our testes cells? So we're synthesizing steroids. Yeah, this would be the smooth ER. So if you remember steroids, they're a type of lipid. Right? So we're not talking about proteins, we're talking about lipids. And it's a smooth ER that synthesizes lipids. Right? Steroids being a type of lipid. What about this last one here with our muscle cells, where we have lots of movement, we have a high energy requirement? I know you all know the answer. I've heard the answer. Mitochondria, thank you. I know. I hate doing that awkward silence, but sometimes if I do it, someone will speak up. Thank you, Hannah. Mitochondria, yes. The powerhouse of the cell. It produces all of that ATP. All right. Any questions about that question there? I really like that question, and it is a type of question that you might see on an exam because it asks you to kind of synthesize material from many different subunits in our lectures here. All right, so we're going to switch our focus now to cell membranes, and we're going to talk about cell membranes today and on Friday. So, we've talked about it a little bit, right? We know it surrounds the cell. We know it's made up of lipids, specifically phospholipids, and it's a phospholipid bilayer. So if we look at our cell membrane here, it's kind of zoomed into our phospholipid bilayer, right? We have these two halves, and our phospholipids are what's making up our membrane. Remember, our phospholipids, they have that hydrophobic tail and the hydrophilic head. So half of our phospholipid is water-loving, half is water-fearing. Right? And our water-fearing tails, our hydrophobic tails, are going to be towards the inside, creating this hydrophobic interior. Now, cell membranes also have a lot of proteins embedded within them. We will talk about different types of those proteins and what their functions are. A lot of them really help regulate transport of material in or out of the cell. So we would call this membrane semi-permeable because it doesn't just allow everything to go in and out, right? It regulates what goes in, what goes out, and the timing of which. Right? Now, I put on here all cells have a cell membrane. Right? I am going to amend that by saying all living cells have a cell membrane. There are examples, especially in plants, where they have cells that perform functions, but the cells are actually dead when they're mature, and they don't have a plasma membrane. So all living cells have a cell membrane, especially all cells you will find in animals. If we're looking at our cell membrane here, we have these two leaflets, right? And they are not identical to each other. So the top half and the bottom half, they are asymmetrical, right? Particularly this top half that touches the extracellular environment here, they may have a lot more proteins embedded for cell-to-cell signaling, right? There's no use in having those proteins on the inside of the cell, right? Because how are you going to signal to another cell if you're just signaling to yourself on the inside? So we'll have a lot of those on the outside. Right? We'll have some cholesterol embedded within our plasma membrane here. We'll talk about why in a second. But they're asymmetrical. All right, we do have a lot of proteins. And we're not going to go over all the proteins, but we are going to talk about the three different types of proteins that you might find. So the first would be a transmembrane protein. Right? This is a protein that spans the entire length, or the entire width, I should say, of the plasma membrane. Right? It has a side, has a side towards the outside of our membrane and has a side towards the inside of our membrane. Right? They are embedded within this hydrophobic layer of our membrane. So we often like to simplify it in pictures and show this like really rugged blob shape for our proteins, all right, but they actually have these very specific regions where the chemistry of our amino acids allows them to embed itself in our hydrophobic layer. Mm-hmm. We also have these lipid anchored proteins. Yes? If we look at this, these regions that are like labeled 1, 2, 3, 4, 5, 6, 7, those are actually all hydrophobic layers because it allows it to mesh with the hydrophobic inside of the plasma membrane. But yeah, it could have some more like polar regions that are more hydrophilic on the ends. Yeah, depending on the specific function of the protein. Yeah, for sure. Any other questions? That was a good question. Thank you, Maddie. All right, so our second type of protein is this lipid-anchored protein, right? It's not embedded in the plasma membrane completely, but it is attached to a lipid that will embed itself in this membrane. So it doesn't span the entire membrane, it just sticks out on half the membrane here, and it's embedded by this lipid. This lipid is covalently attached to one of the amino acids, and it keeps that protein in the membrane. Our last type of protein is a peripheral protein, peripheral membrane protein. They are not directly embedded in the plasma membrane, right? They are either going to be non-covalently bound to another protein, which this is a good example of it being bound to another protein, right? Or it might be bound to the polar head of a phospholipid, so any one of these heads here. But it's not directly embedded, like this transmembrane protein or this lipid anchored protein. Right? So it's non-covalently bound, so a lot of the binding that would be, what we would see here would be like hydrogen binding or ionic binding. Now it's important to mention that these membranes, they're not static. There's actually a lot of movement that happens in our membranes. And so because they are not static, we call them semi-fluid, right? Now we're always going to see our hydrophobic tails towards the interior, right? And our hydrophilic heads towards the exterior, right? We're always going to have that configuration. But these phospholipids, they can have some lateral movement, so they can move within the same plane of our layer. They can have rotational movement, so they might rotate in the same part of the layer that they were originally in. We can, in some cases, it's not as common, right, have a phospholipid that actually does this flip-flop, right? Because it has to bring this polar head through this non-polar region, it has to use an enzyme that we call flipase to help it move from one side to another. And because it needs an enzyme, it's going to use energy. This is very unfavorable for the cell, so we don't see this type of movement a lot. Right? The big picture here is things are constantly moving. Right? I read, I forget the specific value, but in your textbook it said something like it will change positions like 10 to the 7th time every second. So there's a lot of movement. Right? Now, why do our membranes need to be flexible? Well, it allows for it to respond to its environment and change its shape, right? So it can kind of move and be flexible. It also allows for our cells to grow, right? So if we need to add more on, if it was rigid, you'd like physically have to break the membrane, right? But because it's fluid, it can just add more on, create a bigger cell. Everyone's happy. Now, there's different factors that affect how fluid our membrane is, right? So the length of our fatty acid tails of our phospholipid, right, they will affect the fluidity, right? If the tails are shorter, right, they're not going to interact with the tails on the other half of our membrane as well, making the membrane more fluid. So you can kind of think of it, if we had short tails, right, they're not interacting as much, they can be very fluid, right? Versus if they're long tails, they're going to have more interactions with the opposite side. There's going to be a little bit more friction going on here. They're going to get tangled up, not as fluid. Now we know phospholipids, they're made of fatty acids, and some fatty acids can be unsaturated. So when they're unsaturated, they have that double bond, right? The presence of double bonds will indicate the fluidity of the membrane as well, right? So if you think about this double bond, it creates a bend, and when that bend is created, right, it's kind of making that tail essentially shorter in its reach, so it can't interact with the opposite side as well, therefore making it more fluid, right? So the more bends you have, the more unsaturated fatty acids are in the phospholipids, the more fluid our membrane will be. And finally, we have the presence of cholesterol. Right? So we have cholesterol embedded within our membranes. And the importance of this is that cholesterol can stabilize membranes. Right? Now, it's very temperature dependent. Right? So when we have a really high temperature, right, the fluidity can be decreased a little bit. Right? So it's not going to move around as much. The opposite is true when we have low temperatures, that fluidity is going to be increased. And for those low temperatures, if we're increasing the fluidity, increasing the movement of these molecules, that helps prevent these cell membranes from freezing. Right? So cholesterol, they play a pretty crucial role, especially when we get to those low temperatures. Now, one thing I want to add is that proteins, they can also move similar to those phospholipids, right? They can move laterally, they can rotate within our plasma membrane. They're not going to do the flip-flops so much, right? Because they're very large protein, it's harder for them to make that type of movement. But there are some proteins that just, they won't move. They will be static, right? And it's the proteins that might be bound to either that extracellular matrix or the cytoskeleton. Right? So if it's bound to either of those, it's not going to move. Right? That protein will be static. Now the phospholipids around it can certainly still move around that anchored protein, right? But that protein will be anchored in place. Now, there was a pretty cool experiment that kind of showed us that our proteins can move, all right? So this experiment was done in 1970, and their end result was basically that proteins move about 30 to up to 90% of the proteins in our membranes can actually move within that plasma membrane. And so what they did is they took a mouse cell and a human cell, and they fused it together in this hybrid cell, right? Now, I have not read this paper, I've just read a summary of this experiment, and I'm assuming they fused it together because it looks like these mouse cells are more readily able to attach what they did, which was a fluorescent dye, right? So they attached fluorescent dye to those mouse proteins in the plasma membrane, right? That allows them to kind of track the movement of those proteins. They then heated up our cell, put it in an environment that was about 37 degrees Celsius, right? That allowed for more fluidity, right? And when they cooled it back down, so it was stable, they saw that yes, the proteins moved, right? They did not stay as they were originally saw them, right? They moved all over the place. Right? So that was a pretty cool experiment. I like kind of seeing the science behind why we know things happen. All right, our last group question here. Right, I want you to think about this fluid mosaic model. Right, so we call the fluidity of our cell membranes the fluid mosaic model. Why is it called that? Right, think about those definitions of why is it fluid, why is it mosaic? But not all of them. I'm thinking like mosaic. Yeah. I will take that and we'll think about that. All right, so why do we call it the fluid mosaic model? Why do we call it fluid? Because it moves, yes, there's movement involved. What about, why do we call it mosaic? What was that? I didn't quite hear you. It all goes together. What's all? What would be included in that all goes together? What comprises a phospholipid membrane? We have the phospholipid bilayer. What do we also have within our phospholipid bilayer? Yeah, we have many different proteins with many different functions. It's a mosaic of things coming together. Yes. All right, before I let you leave here, I do want to introduce another assignment that I'm going to have you do. Right? This is going to be due a week from today, so you'll have some time to do it. And hopefully you'll find it interesting. Right? It's about scientific literacy, like what does a paper publish, and then what might a media report about that paper. Right? I think it's a fun assignment. I hope you think it's a fun assignment. I will have a submission site. I think I actually have a submission site already up on Moodle for this. Whenever you are done with it, you can submit it. We will discuss it next Wednesday. All right, for class on Friday, I want to try something where we go outside. So I am asking you to watch a pre-lecture that I have already recorded for you. That material is fair game for the exam for sure. And I will give you a chance to ask me questions about that material at the beginning of class on Friday. But I'm hoping we can get us outside. I don't know.